Hello, I'm going to introduce the topic of mitosis, and it starts off with well, let's first off, what is mitosis? Mitosis is a type of cell division that results in two identical daughter cells from one parent cell. In interphase, this is a period of cell growth when the cell makes new molecules and organelles. Chromosomes are thin and not condensed yet. In prophase, in this phase, changes happen to chromosomes to make to make them the chromatin fibers coil. Um, the nucleus for chromosomes become thick. They exist as two identical sister chromatids joined together at the nucleus region, called centromere. The spindle fibers begin to form, driving breaks. The nuclear envelope begins to break. Fibers contact the centromere of the chromosomes. Chromosomes move closer to the center of the cell. In metaphase, the condensed chromosomes align. They align in the center. The meiotic spindle is completely formed. All the chromosomes centromeres line up in the middle. Two poles of the spindle. Okay, they align between the two poles. The tracks of the meiotic spindle connect to the sister chromatids, and they tug towards opposing poles, as you see, and this keeps the chromosomes in the center of the cell. In anaphase, chromosomes split and move closer to the center of the cell. As you see, right here, they're moving closer to the cell. And usually, cleavage furrow forms, which is an indentation that forms on the surface of an animal cell as it begins to divide during the final stages of cell division, mainly in anaphase, telophase, and cytokinesis. During this phase, also separation of chromosomes. There's separation of chromosomes in this phase. The sister chromatids separate, resulting in two unconnected chromosomes. Then the chromosomes in the cell move away to opposite poles as the spindle tracks shorten in the process. At the same time, the chromosomes not yet connected to the poles get pushed farther apart and the cell lengthens. One of the final stages of mitosis, the cell divides, producing two identical daughter cells. In telophase, the cells form and along with cleavage furrow. And you see, this is the cleavage furrow, the indentation you see. Now each group of chromosomes and groove are put up at opposite ends of the cell. Essentially, what happens in the phase is a reversal of prophase in a nuclear envelope form, uncoiling the chromosomes. Membranes that surround the nucleus form each chromosome. The chromosomes return to a much less condensed, almost like thread look-alike. The spindle fibers disappear. Now this division of the nucleus into two identical daughter cells or nuclei is done. Along with telophase, cytokinesis occurs division of cytoplasm. A cleavage furrow pinches the cell into that producing two daughter cells. This happens in animals and in humans as well, and in non-animals it's different. Plants do not form a cleavage Furrow, they build a new structure, the cell plate. Humans do form a cleavage furrow during cell division. The furrow happens by a contractile ring of actin and myosin proteins which tighten near the cell's center, which pushes the membrane inward, leading to the cell being sort of pinched in that results in two identical daughter cells. The furrow is important for the separation of the cytoplasm and finishing cell division. For nuclear proper cellular contents and its completion of mitosis.